the other thing that I'm very, very happy about as I get to sharing my screen is that this is luckily scheduled at a time when I'm in Europe. So it is 1 a.m. Local time in Sydney. So thankfully, this is like a normal, a very reasonable 3 p.m. local time. I think for this seminar, I'm very happy for people to use like, the raise hand or like, just jump in with any questions. So if it sort of becomes a little bit more of a discussion or, something like that, then I'm very, very happy for that. So yeah, with that, I'd like to talk about a topic that has become, quite an interest to me over the last sort of 1-2 years or so, and that's Mosaic single cell data integration. So I'm sort of including like my postdoc affiliations here as well as my current Sydney affiliations, because I feel like this project is very much like a bridge between these two projects that I've been looking at sort of as I transition into my new research program. So maybe a bit of context around what I did during my postdoc. So let me get into my slides here. So in our lab, we were very much interested in this topic of early organogenesis in mice. And so what you can see on the left here is this sort of schematic of what happens to mice as they develop over these sort of 3 key, very eventful days of their development. We sort of start from this embryonic day 6.5, where the mouse embryo is very much a relatively simple sort of 3-layer structure. And then over these, 3 days, there's the stages of gastrulation and early organogenesis. So you can see that in terms of the schematic, there's a massive change in terms of scale and in terms of the types of cells that arise. So, we go from this sort of simple structure into, something that almost resembles kind of like an organism. We have like this body plan, as well as, something that may sort of elongate into forming your organs. So the lab, is interested in studying the molecular profiles of these tissues at the single cell level. And so what you can see along this slide here is a UMAP representation where each point corresponds to a single cell. And as the animation is going along, we have different time points across this developmental stage being highlighted. And so, yeah, what we can see is that there's this sort of massive diversification of the cell types that are emerging. And there is quite a lot of sort of variation in terms of the gene expression profiles that we're seeing across this time. But the thing to remember is that, this UMAP representation, this doesn't necessarily reflect, what's going on in terms of space itself. So this is where my postdoc project came in. And, we wanted to very much, understand, cells in time, but cells in space. And so this was a work that we did in collaboration with Professor Long Kai in Caltech. And this was joint with a fantastic PhD student who's now graduated, Tim Lohoff at the Babraham, as well as some wonderful collaborators as well that you can see here. So this was a consortium effort to start to characterize the molecular profiles of mouse embryo in space. And so what we did was look at whole organism sagittal sections at the stage roughly E8.5. So that's that later stage that I showed in the picture just a bit earlier. And so what we captured using this technology, SeqFish, was a whole organism, sort of multi-scale capture of gene expression in space. So what we can see on the first panel here, this is a digital in situ, so a reconstruction of the molecules that we observe for a given sort of subset of genes. And so if we zoom in, then we can see that, we can see a little bit more structure at the tissue level. So we're still seeing those same molecules in space, but we can start to sort of resolve a little bit of our cell information as well. And if we zoom in once more, then we can see that, yes, indeed, we are able to capture sort of molecule localization in space, as well as individual cell segments. So I guess the thing to sort of note for this is that, we were able to capture, a whole suite of genes. So we captured 351 barcoded genes all together in this seqfish panel in the same coordinate space. And I guess on top of that, we had quite a scale in terms of the number of kind of like the spatial features that we may be able to extract from this. And I think that this is something that maybe we could sort of keep capitalizing on a little bit more, as a field, I guess. So now for this particular work, one of our key aims was to perform an integration of our seqfish data with our pre-existing dissociated single-cell RNA-seq data set. And so this is where this question of, data integration really comes in. So for us, our goal was to take our seqfish data set and be able to annotate it using essentially the vocabulary that we had for the well-curated dissociated data set that we had. So, we can see our UMAP representation of our dissociated data. Now note, this was a whole transcriptome study. So we've got all 20,000 genes, right? And in our seqfish study, we have, our set of, like well-curated, but certainly not the whole transcriptome set of genes. And, I'm sort of showing like the UMAP representation of these cells in space, but recall that, these cells also have their corresponding physical coordinates, and they're in gray because we haven't yet annotated them. And so how did we do that? Well, we restricted to overlapping genes, and then we used a data integration technique called mutual nearest neighbors to embed them into a common low-dimensional space. So what I'm showing here is a UMAP representation of this low-dimensional space, and this is joint between the two. So we can see that the colors correspond to the different data sets, and overall we're seeing quite a good overlap between these two. And we would hope that would be the case, given that these are wild type embryos, and we would expect them to be comparable between each other. So, that's the way that we do the integration. So what can we gain from that? Based on this common embedding, then we're able to use essentially traditional machine learning. So something very, not so sophisticated, but thankfully quite robust, cell type classification, just using K nearest neighbors. And so, if we split this out up on the top, we can see, the top part is our original cell type labeling using our dissociated data. And then from the bottom, we can see that this is our predicted cell labels for our seqfish data. So that's our predictions. And so what does that give us in the end? It allows us to essentially take our, our curated set of cell types and their characteristics and so on, and be able to basically paint this onto our spatial reference, and so onto our physical coordinate space. And so this was quite helpful for us because it allowed us to be able to characterize some things about the spatial distribution of particular cell types in space. So, if we look, for example, at, the brain region here, so like these dark green up in the top, we see that this is, it's actually covering a very large area of the overall space. And whereas in the original UMAP space, you know, it's not necessarily covering so much of this area here. So that I think was quite helpful for trying to, you know, be able to get into this common framework for downstream analysis. Are there any questions at this point or anything like that? No, I keep going. Do feel free to jump in. But I think the thing that, you know, really sort of got me thinking more about, you know, this was, maybe I'll go back this slide, was it kind of didn't sit so right with me that, you know, we were kind of throwing away that like 19,650 genes like that, you know, we're sort of not capitalizing on so much. And so, you know, it really was like during this project, I think that, you know, for this particular data set, we did capture quite robust, you know, sets of quite a robust mapping along. But, you know, kind of, I was thinking, like, what if we could do more? Like, could there be like a better way to do this? And what if we didn't necessarily have that, you know, great sort of set of genes that best recapitulate the cell type as well? So that's a little bit of foreshadowing for the rest of the talk. But I want to step back a little bit and, you know, talk about, you know, what we mean when we say data integration. And I'd say that this review sort of perspective piece that came out of the lab is a really great way of sort of characterizing exactly what we're doing when we say data integration, because it does encompass a lot of different you know, topics all in the one word. So I guess, you know, we can think about it in terms of 4 different types of integration. So the first is this idea of horizontal integration. So what you're seeing here is sort of a schematic where we have these populations of cells. So you can see these are our populations of cells, and with these populations, we've measured sort of the same omics modality, or we've measured the same set of features. So in this case, this is like polyadenylated mRNA. So we're capturing something like, you know, single cell RNA-seq from this set. And then overall, you know, what this corresponds to is this notion of horizontal integration. And we get this idea of horizontal because we take our data and we sort of project that we represent it as, you know, rows corresponding to features and columns corresponding to cells. So you can see that we have the common set of features across multiple distinct sets of cells, right? So that's horizontal integration. Now, contrast that with this notion of vertical integration, right? So this is an idea where, say, if we've got like the same population of, say, cells or samples or something, and we're able to capture multiple different sets of features or different modalities, but from the exact same cells, right? So we've got the same cells with multiple distinct features. So here, you know, we've got like DNA methylation, we've got our RNA, and we have chromatin accessibility as well. Excuse me. And so here, you know, we're calling this vertical integration because we have the same cells and we're capturing multiple sets of features. So that's, you know, the sort of 2, I'd say, most well-studied sort of flavors of data integration up until this point. So, you know, if we think about, you know, what are examples of, say, horizontal integration, you would think of things like such as, you know, what could be called as like batch correction techniques. So an example of horizontal integration, and I already talked about this in our seq-fish single cell RNA-seq integration, is this method called mutual nearest neighbors. And so the idea is that you have multiple sets of cells, you've captured the same set of features, and now you want to be able to align them into a common space by, you know, making use of these sort of individual pairs of mutual nearest neighbors, right? So that's to give an example of a horizontal integration approach. There is another example of a horizontal integration approach. So it can go sort of as sort of, having as much prior knowledge as you want, or as least. So this is an example of a horizontal integration method where you have actually some prior knowledge informed as well. And that's in the form of these stably expressed genes. So you can do all of this batch correction to sort of bring about. So that's two examples of horizontal integration. Now, examples of vertical integration. One would be this approach called MOFA, multi-omics factor analysis, that came from a colleague of mine from John and Ollie Siegel's group. So the idea here is that, you have these sets of features that are common across samples, and then you're able to sort of jointly embed them into a common space by taking into account the individual sort of data distributions of each of the omics. So, things like DNA methylation, the distribution of DNA methylation is not necessarily the same as gene expression, and so on. So you may want to be able to perform the factor analysis sort of distinctly, and then be able to bring these in together. Another approach that is an example of vertical integration, which you may be familiar with, is this approach called weighted nearest neighbors, coming from Rahul Satij's group. And so this idea is that, you have data from multimodal data technology. So, you have different combinations of omics happening on the same cells, and then you're able to sort of place them onto the same common embedding, using this weighting technique to give like an equal weighting towards a common low dimensional embedding. Right, so that's another example of vertical integration. Cool. So, that's great, but my title is about Mosaic data integration, so maybe I'll try to get to that as quick as I can. And I'd say that the motivation for this is coming from like two things that I've seen coming in our sort of research community. And the first is, we're seeing an increase in the number of studies of like single cell transcriptomics. So, this is a plot of the time, so years versus the number of studies that are coming out in single cell transcriptomics. So there's that. But then in addition, we're also seeing an increase in the variety of different types of single cell technologies. And with that is an increase in the variety of the features that we're actually extracting from the resulting data sets. So along the right here, you can see this schematic of a cell. And, these are all the different types of omics that we're able to extract from cells at present. And probably there's a bunch more as well. So, we can characterize the DNA, we can characterize the RNA, we can characterize the protein, spatial positions, lineage, and so much more. And the idea there is that, as we increase the number of technologies that we're capturing, and as we want to be able to combine them, what you may find is that, that set of intersecting features just becomes sort of vanishingly small, right? So as you sort of restrict down to those common set of features, genes, and so on, you may actually end up with nothing. So you kind of may want to sort of work more in a sort of more forgiving space where we have more features. All right, so here's the corresponding schematic of Mosaic single cell data integration. And here it's a little bit of a Wild West, right? So we've got some situations where we have, this kind of sets of cells where we have like the same omics being performed. And then, we have some examples of like multi-omics as well. So the resulting, landscape of this data sets becomes very complex very quick. So, here we've got an example of like a set of cells where we have captured 3 different sets of features. Here we've captured 2 different sets, and so on and so on. And so you can see that, if we wanted to, perform, say, a vertical integration or a horizontal integration, it's just not clear how exactly you would do that, right? And so, our goals for trying to address this kind of problem, is a method. We wanted to develop a method that performs mosaic data integration and be able to use whatever information that we can gain from those non-intersecting features. So from those features that are not common across all of the different data sets. We want to ensure that we can, enable indirect mosaic integration. So we want to be able to kind of connect multiple data sets to each other in some way. And, finally, we want some way to be able to incorporate prior information. So, for example, cell labels within our data integration. So with that, I guess, the thing that we want to, also consider is, why exactly would we want to perform this integration? Now, I showed you an example just before that, we performed an integration to get to a joint embedding where we could perform downstream machine learning so that we could perform label transfer. So, other things that you can do, there's the benefit of joint visualization, so yes, joint supervised learning. We can do joint unsupervised learning, so we could find common clusters and subclusters that we can then characterize, using some information that we may have from one data set versus another. We could perform, say, something like cell abundance-based hypothesis testing. So, we could ask like which cell types might be enriched or depleted within this common space. So we could use, this embedding to perform point imputation. So we may want to sort of try to understand what features might we have measured if we had the capability of doing so. And as well as, just sort of joint bespoke analysis. So things like pseudo time or, velocity or like, any kind of typical kind of technique that we may want to be using depending on our biological questions of interest. Cool. So the technique that I've developed is called StabMap. So it's called that because it's aiming to stabilize mosaic single cell data integration. And it aims to do so by using these unshared features. So there's 3 steps in a StabMap technique. The first is to identify mosaic data topology. Second is to build reference embeddings for chosen references. And then third is to project all non-reference cells onto that space by traversing the shortest paths along this topology. Right, so let me give a bit of an example, like a schematic here. So here we've got an example of, our observed data matrices. So we've got our cells by our features, and we can see that there is some degree of overlap between these data sets, but not a full overlap, right? So from these data, we can see that there are 3 data sets. We extract out a mosaic data topology. And so this is a graph where each data set corresponds to a node in the graph. And there is an edge drawn between these nodes if they share features within their data sets. So you can see that, we've got an edge drawn between the red and the blue data set because they share some features. But we can see that there's no shared features between the red and the yellow data set up in the top here. Right. And our goal is to essentially estimate a common low dimensional embedding where we have the combined coordinates by the cells. So we want to be able to get to this space here. And note that once we do get to this space, we're in a position where we could perform sort of a downstream horizontal data integration if we so chose. Right. So, we can enable a mosaic integration followed by a horizontal integration if we would like to. So so here, if we've got, a particular reference data set that we choose, so here we've selected the red reference data set, what do? We take our query and we want to be able to project onto the low dimensional coordinates as described from our reference data set. So you can see that these axes here are sort of red, and so I'm trying to convey that this is as learnt from the reference data set itself. And we do that using a linear model framework. So we treat the response, so the outcome of our linear model, as the reference coordinate scores themselves. And then our predictors are what are the shared feature values, right? So how much can we recapitulate the scores based on those shared features? And once we build that model, then we can simply project our new data onto that space, right? So that's fine. But then I think what's kind of nice is that, we can create an iterative process to be able to do this. So if I have another query data set, and now I'm essentially treating the path, the data set along the path as an intermediate, right? So now I'm going to fit a new model where I have the response being the scores or the predictors of the intermediate data set. So then I can predict onto this space, and then using the previous model, I can just simply project along again. And so the end result there is that, we have a single embedding for these multiple data sets into our reference coordinate space, right? Now, the thing to note is that, here we selected this data set as a reference, but we can do this multiple times with multiple different references being chosen, right? So if we had our blue data set, say, being selected as our reference data set, then we could select this, and so on with the yellow. So then we can combine these by performing a reweighting of our low dimensional subspaces, right? So then our final combined coordinates would be sort of essentially like a weighted mixture of the scores from our set of references all up. Yeah. Okay, so I mean that's what StabMap does. So it's I guess I can give you an example of how you would do it. It is available as a preprint at present and is on GitHub as an R package. So a little bit of what the code would look like if you were to run it. So here we have essentially a list object where we have the feature matrices for our data. So this example here, I've basically extracted the PBMC 10x multiome dataset from the 10x Genomics website. So this is a dataset where in the same cells, we have a single cell RNA-seq and ATAC being performed. So we've captured gene expression as well as chromatin accessibility, right? And so for this example data, what I do is I sort of create this situation where imagine we had our multi-omic data set, and then imagine we had single cell RNA-seq and then single cell ATAC-seq as well. So here, if we're looking at what is the dimensions of these, these are sort of about 3,300-ish simulated cells with different sets of features. So this will be the genes for the ATAC. These will be like selected peaks corresponding to different regions of the chromatin, and then the joint space between them for the multiome. Right, and what is this? So this is like a sparse matrix representation. You could input, say, something like a single cell experiment or summarized experiment as well, if you so chose. So, you can examine what kind of information we actually have among this sort of set of data. So here we have a mosaic data upset plot. Here we've got our three sort of modalities. We can see that what is common in terms of features is between the RNA and the multiome, and then the multiome and the attack. So, we can see that there's actually no common features between the attack peaks and the RNA. And we've sort of made it so we haven't performed any sort of extra peak matching with like promoter regions or anything like that. We have not done that. So on to that, if we wanted to think, what is the mosaic data topology of this sort of set, this list of data objects, we can extract that out. This is an iGraph object. So you can see that it has 3 nodes, 2 edges, and it's characterizing this relationship between the data sets there. Now, if I want to integrate these data, then I would be using, excuse me, the StabMap function call, right? So in goes the list of data, and then we specify exactly which we want as our reference. So in this case, it might make sense for us to use the data set that captured the most number of features as our reference. But, it may be that it's sort of worthwhile to think, like maybe the RNA was better characterized in terms of cell types or is more broadly sampled. So maybe that might be a different thing to consider. So yeah, so then from that we have sort of generated embeddings and then being able to traverse these paths. And then we have, in the end, a common embedding between them. So yeah, then following on to that, we can essentially treat this as if we'd perform something like a PCA, right? So we can perform UMAP, further low dimensionality reduction. So here we've colored the cells here based on the data type. So this is RNA, multiome, and attack. And we can color it by the cell types as well. So we can see that, yes, we are able to, recapture the cell types, even though, we threw out this sort of extra information in our simulation. Cool. Am I going for time? I think I'm okay. Are there any questions at this point?
Maybe one small question, or rather just wondering,
Yeah.
When you do this mapping multiple times, as you showed, like for data sets that have no overlapping features at all, and you go through this like intermediate data set that are kind of like coupling the 2 data sets, are there any drawbacks to do this like multiple times as opposed to when you have them connected to a certain extent? Just wondering, because like mapping problems in bioinformatics are typically lossy. That's why I wonder if you do this multiple times, whether there are any drawbacks to that.
Yeah. So it's this thing that, if you keep mapping sequentially, you are going to sort of lose out to sort of stochastic variability, right? Like there's, you are fitting a model, so you have some kind of noise that is going to be captured, and then you're going to sort of try to propagate that. We've done some sort of stress testing, I guess, for this integration. And I mean, what I said was that this is a very flexible approach, right? Like you can sort of chain up as many data sets as you like. I mean, whether or not it's good too, that's a different story, but the code works if you sort of, if you chose like 10 or 15 data sets. So we did do a simulation where we took, I think it was up to 8 data sets, and then we saw how like, what does that loss curve look like? And we did find that it kind of plateaus. So after about 2 or 3 steps, you see the biggest sort of loss, and then it's just sort of plateauing. But yeah, it's a concern. I will have a slide on the like underlying assumptions in a minute.
Interesting. So the baseline is if there are certain overlapping features, you end up in typically a better integration. Is that a fair statement?
Yes. I mean, yeah, I guess that's the thing. Like in these integration spaces, it's not going to do magic, right? You need there to be some kind of information for it to be preserved. So we can do our best to try to keep it. But if it's not there in the first place, good luck.
Excellent. Yeah. Excellent. And we also have another question from Robert at this point.
Yeah, thanks, Ludwig. Hi, Sheila. Sorry I was a few minutes late. But I have a feeling I saw much of this a couple of weeks ago. So my question is, so it's sort of nice to think of the features as abstract things, but they sort of aren't abstract things. And so to what extent have you and the others, you know, sort of thought about annotations on features, right? So there's different functions that RNA take on. So overlapping, you know, things that have to do with, let's say, DNA replication is really a pretty different thing than having overlap of anything involved in sort of metabolic activity or cell signaling and so just sort of curious about, and then, you know, if we would go to site-seq and these other things, it's very similar sorts of things, that there seems to be a lot of information in the annotation, right? The functional part for these different quantities. And you know, have you thought about how that would come into this game?
Yeah, it's interesting, and it's something that I've only just implemented like recently with the software itself. So, you know, I said that there was this like reference embedding, right? Like this sort of goal embedding that we have. Now you can imagine, like, so if you run this code sort of by default, it's going to perform principal components analysis, or it's going to perform linear discriminant analysis if you've given labels. But you can imagine that, you know, like you said, there's these sort of sets of features that are relevant in their own way, and they may warrant, you know, something like a vertical integration approach for getting at a low dimensional representation. So imagine something like running MOFA, where, you know, you have the RNA modality and then like the protein modality and so on and so on. So what I've implemented now is, you know, you can have your own target sort of set of reference coordinates. So say this could be the output of, say, MOFA that has taken into account the different sets of features. But the thing that StabMap does is it's still going to use all available predictors to try to get to that response, right? So it is going to treat the input features equally, right? Like it'll just be a linear model. So I think like in this vocabulary, it's like you could perform, excuse me, like a vertical integration followed by a mosaic integration, potentially followed by a horizontal integration to you know, get to the biological question. But yeah, it's a great point. Cool. Thanks for these questions, and I hope that it sort of, you know, helps with a bit of helps a bit with the rest of this seminar. So yeah, so, you know, what are the requirements, the features, and like the sort of underlying assumptions that we make with StabMap? So I did say that there were 4 flavors of data integration. So we had, you know, the 3, but then there's this elusive 4th diagonal. So StabMap does not perform diagonal integration. You need the mosaic data topology to be connected. So there need to be paths going between the different data sets. The other thing in terms of features is that it's implemented in a general manner. So, you know, it can be for any connected topology. So you can input a list of any size or a set of single cell experiments of any length. There are user-based weighting for the reference coordinates. So maybe you have 2 different data sets and you don't necessarily want to give them 50-50 contribution. Maybe you want to upweight one set of data a little bit more than another. The other thing is that it's deterministic and linear. So it's pretty quick to run. And I guess the other thing is that because we are fitting linear models, right, you know, it does require some sort of data set normalization, so some scaling and centering of the data. Now, this last point, it's only as strong as its weakest link, is a bit of this sort of thing that I hinted with Ludwig's question about, you know, how does it relate as you go across? And so I guess we can kind of have a bit of, excuse me, like intuition there that, you know, if we have this information that's being captured, you know, we need to have enough transfer across different data sets. So if we have, you know, one intermediate data set that has no information or non-informative features, or, you know, random cells that, you know, are not captured, then you know, we're not necessarily doing a good job. So additional assumptions that we have are that there's, you know, no confounding of, say, the biological signal between the data sets, and that there's enough biological information that's captured among the shared features. So for this point here, you know, this I guess is very much capturing this idea that if we have a reference space, we want to be able to have enough variation captured so that we can perform that mapping onto a reference space. Right. So in the remaining sort of 10 or so minutes, I'd like to go through a case study. So we did do some simulations and, you know, benchmarking and so on. That's in the preprint, but I like to sort of go through this case study because I think it's quite cool. So I'd like to share. So in our lab and with our collaborators in Cambridge, they had performed a chimera experiment using single-cell RNA-seq. And so what did they do? They took mouse embryonic stem cells that had a knockout of a gene brachyury, so this T, and those cells were injected into a mouse embryo and then allowed to sort of develop as normal. And so then this results in this chimeric embryo, which can then be sort of dissociated, FACS sorted, and then subjected to single-cell RNA-seq. And the idea here is that, you know, we have the one organism, but we've got different, you know, cells that are wild type and mutant within this same organism. And we like to understand, you know, are there any changes in terms of the cell types that emerge as they develop over this time? So here, you know, I said earlier that we had our spatial reference map of mouse embryos. And so here our integration question becomes, you know, here we've got our single cell RNA-seq. We have genes that are sort of covered in our seqFISH library. We have genes that are not covered in our library. And then we also have, you know, additional features that we could extract from space itself. Right, so here we've got the expression of the nearest cells in physical space. And you can imagine that, you know, if we had captured the single cell RNA-seq without needing to dissociate, we would have been able to capture this information as well. So this essentially is like a latent set of variables that, you know, we're not getting there. So what we did was take these data sets, so our dissociated perturbation experiment, as well as our wild type spatial atlas. And then we performed StabMap. So we embedded them into this joint space. So this is a UMAP generated from this space. On the left, we've got the cells. So these are a mixture of cells colored by the cell types. So we can see that, you know, we were able to discriminate between cell types. But then if we look on the right, you know, we have our nice overlap between our dissociated data and our seqFISH data. So we're seeing that that's quite consistent. So, I mean, remember the dissociated data actually has perturbation information associated with it, right? So for each of the single cell RNA-seq cells, we actually have a label. Is it a wild type or is it a knockout cell? And so what we could do from that is perform a local enrichment test. And so we could ask within this low dimensional space, if we identify, small neighborhoods, what level of sort of imbalance do we see of the wild type versus the mutant cells? And so these are the cell types, and the colors correspond to our enrichment coefficient, right? So we have a bit of a mixture going on across here. And I mean, some of these things, we found that were quite highly consistent with what was already known and what was previously published. So things like the somites, we found that, there was an enrichment of these knockout cells over in the sort of Semitic tissues. But I want to highlight this one cell type here, the splanchnic mesoderm. And what was interesting was that because we have this local test, and because we have a low dimensional embedding that is common to these cells, we were able to perform essentially a label transfer to get a local prediction of the enrichment coefficient itself. Right, and so if I subset down to this splanchnic mesoderm, so splanchnic mesoderm is a subset of cells that will sort of develop to form the tissues that sort of coat your internal organs, so very important. And so what I've done here is plotted the corresponding cells and colored them by their predicted enrichment coefficient. And so hopefully the arrows also help sort of note that what we see is a shift from enrichment to depletion as we go from the anterior, so the top, to the posterior axis of the embryo body, right? And so this kind of is, I think it's a nice observation that we can make, or a nice sort of hypothesis that we can, generate, that we don't necessarily need to have performed spatial perturbation, right? Like we can perform the dissociated perturbation followed by our spatial reference. And by virtue of performing this joint embedding, we can then sort of get our best guess at what may be happening in terms of the perturbation itself. So I think that's cool. I kind of I think it's nice that, we can use sort of the strengths of both technologies and then sort of bring them together in silico. So now, of course, this approach, I think this is sort of like technique overall of Mosaic data integration. It's become quite popular, and I'm very happy about that, because it means that I'm not just in this like corner of research on my own. Like thankfully it seems like it's an important problem that people want to tackle. So there are other techniques. So there's these two that recently came out in bioRxiv, but I do want to highlight sort of three others that do have I think it just shows how complementary the approaches are for performing mosaic integration, sort of maybe distinct from, say, like horizontal or vertical integration. So one is this approach called UINMF. So this is unshared features, non-negative matrix factorization. And the idea here is that if you have different sets of data with different sets of features, so here in this one example, we've got Z1 features and Z2 features that are not shared between these two, then what you can do is incorporate a sort of assumed to be shared matrix within the factorization, so that it becomes essentially a joint optimization. And so then these authors, were able to perform this mosaic integration using different sorts of omics modalities, so things like chromatin, spatial, and single-cell RNA-seq. So I think that's quite nice. Another approach is called multi-map. So this is a graph-based approach. So the idea is, you have different sets of single cells that have come from different modalities. Then what you're doing is placing them into a graph structure. And the assumption for the graph structure is that it should be uniformly distributed. So then using this, optimization approach to sort of minimize and get as best a uniform structure as possible, you result in an output graph as well as a 2D sort of UMAP equivalent. So this is a multi-map approach. And then finally, an approach that came out sort of earlier this year is excuse me, called bridge integration. So this idea is, using this idea of dictionary learning. So rather than, considering features to be shared, you assume that there exists some cells that are consistent between the two different data sets. So here you have a multi-omic bridge. So for example, any of these approaches, and then you have your single cell RNA-seq reference, and then your sort of single omic query data set can be mapped onto your RNA reference by identifying these dictionary cells. So I think it is quite interesting how creative, I guess, the like overall research community is for trying to tackle this problem. I think that's quite good. So just to summarize, I just want to revisit the goals of the integration. It was to develop a technique that uses these non-intersecting features, enable this indirect sort of idea by taking the Mosaic data topology, so sort of abstracting into a graph, as well as incorporating, say, prior information using cell labels. So like I said just a bit earlier, we could use this together with other sort of bespoke embedding techniques, say something like MOFA, by giving scores a priori as well. So with that, I'd like to thank the wonderful people who have been involved in the projects over these times. I will sort of plug a little bit to say that my group is up and running. It's been 5 months since I started, and I'm currently recruiting a PhD student as well. So definitely happy to have a chat with anyone who's interested in these kinds of analytical problems.
Excellent. Thank you so much, Sheila, for introducing us to this world of single cell data integration. I think it was really, really great to see what kind of techniques are out there and what you developed. We have some 4 minutes left, so maybe I check in with the audience whether there are any questions for Sheila. And maybe I take the start. Sheila, as you know, we are very interested, of course, also in analysis infrastructure for this spatial data. I wonder whether you could talk a little bit more with regard to the anatomy of this SeqFish data set, what kind of infrastructure you use there. I wonder, I'm not too familiar with SeqFish. As you know, we use more of this MERFish data. Do you also have their images? Do you also have this problem of these big images there, or were these rather like you end up with like coordinates and plain numeric data? And I also saw some polygons, so there seemed to be some segmentation involved. I wonder what was used there for segmentation. Was this image-based segmentation, or so it would be very interesting if you could give a look behind the scenes there.
No, I mean, it's quite similar in that, you have this image-based feature extraction, right? And I think the sort of analytical thing to consider is that, when you are performing, say, data integration, you need to consider what is the unit of observation, right? Like, what is it that you're actually combining? Like, is it regions? Is it tissues? Is it patients? And in this case, I'm talking about cells. And so, getting to a place where you can characterize features per cell is the space that we're in. And, you know, the reason why is because so often we're performing this integration with dissociated single cell, you know, usually RNA-seq data. But that need not be the case, right? Like say I had, you know, multiple Murfish data sets or seqfish data sets where, you know, I had different sets of genes being profiled, right? Like this would still be a Mosaic data integration problem, right? And so you need not consider, say, a cell as the unit. It may be a tissue, right? Or like a region or a patient or a sample. Yeah, so I guess with that, the task at hand, because we wanted to integrate with single cells, was to be able to abstract to the level of single cells, right? So we had molecules associated with cells, and then we sum them up to get a gene expression vector, right? For our spatial features, right, we take the gene expression of neighboring features, but then we always sort of calculate that again with a single reference cell in mind. So yeah, there's that thing to consider. Yeah, I think as soon as you get away from like unit of observation is a cell, like there's more possibilities, I guess, for analysis.
And these 350 genes, is this kind of like the throughput that typical seqfish data sets are at now, or what is the scale there? I guess this is some sort of panel that people select beforehand here. These are kind of like early development markers, I guess, that people carefully curated. Yeah.
Yeah, that's right. So yeah, so we had the privilege of having access to the dissociated data before performing the spatial analyses or the spatial experiments. And so that meant that we could identify, you know, the set of genes that is best able to distinguish between cell type classes, right? And so, you know, within the realm of like constraints of, you know, maximum expression and so on. I mean, this work sort of started sort of as my postdoc started. So I think things have progressed since then. But yeah, this is the protocol of intron seqfish. So with that level of scale of features.
Okay, great. Then I think we also have some comments in the chat that people really like to thank you for your great presentation, Sheila.
Thank you very
and
much.
Also,
thanks,
Niels Galenborg said if you want to do some more on the visualization piece, feel free to reach out, which I highly recommend to do. Other than that, thank you so much for coming and this really great presentation. And yeah, I guess we will continue the discussion one way or another on these data sets anyhow. So thank you very much.
Sure. Thank you very much. See you.
Bye. Take care.